All right, so today we're going to be talking about DNA, bacteria, and plasmids. Um, have you gotten to learn this yet? I won't go over the whole thing again, but today we're mostly going to be talking about DNA, getting into some of the specifics of DNA and some stuff we're going to do with that this week. All right, so when we look at DNA, we have what's called a double-stranded anti-parallel structure. Okay, it's overall also a helical structure. And what that means is we have two individual strands, and you can see along here, there's two individual strands. Um, and in this representation as well, there's two individual strands. Those individual strands are molecules that are held together with hydrogen bonds. Okay, so I don't want to get into all the details of chemistry, but basically covalent bonds are really strong bonds that hold together most molecules. But hydrogen bonds are a weaker kind of bond that can cause chemicals to want to attract. It's not really a permanent linkage, but there's a pretty strong attraction here. And so each individual strand of DNA is a single molecule, and they're very long molecules that are in humans up to three. Well, humans are divided into multiple segments, but all of the human DNA is essentially 3.2 billion base pairs. The E. coli genome, so the bacteria that we're going to be working with, is actually a single piece of DNA, and it's 4.6 million base pairs long. And so when we say base pairs, it's each one of these things, these structures here are called a base. And when we have them one next to the other like this is called a base pair. Okay. So our four individual bases are adenine, thymine, guanine, and cytosine. And their specific arrangements and how those bases compare with one another. So if we have an A on one strand, we're always going to have a T on the opposite strand. And if we have a G on one strand, we're always going to have a C on the other strand. And as you can see here, it's kind of random as to which base is going to be in which position. That's sort of life, and we'll talk about sort of how information in DNA is decoded as we go on through the semester. Um, and when we say anti-parallel, if we look at this structure along the outer edges here, this is called the DNA backbone. So these are the bases, and this is the backbone. And if we look at the backbone, it's made up of a molecule called deoxyribose. Okay, so I have that shown here. And if we look at this oxygen in the middle of the deoxyribose, when you don't see an individual corner um, with an atomic symbol, so you can see all these oxygens, you can see a phosphate, you can see nitrogens. If you don't see anything, that means it's carbon. Okay, so we have these ring structures. That's a carbon there, and it's a carbon there, and it's a carbon there, and that's a carbon there, and that's a carbon there. And so there's an oxygen here in the middle of this ring structure in this deoxyribose. And you can see in this representation, all of these oxygens are pointing the same way. They're all pointing up. And over on this side, you can see all these oxygens are pointed down. And that's what we're talking about, the anti-parallel. One DNA backbone is pointing one way, and the other DNA backbone is pointing the other way. If we look a little bit more specifically in that, we call that orientation 5' to 3'. Okay, so if we look at the structure of deoxyribose all by itself, we can number each of these carbons. So this is the first carbon, this is the 1' carbon, 2' carbon, 3' carbon, 4' carbon, and 5' carbon. And so when we're looking at a DNA strand like this, a deoxyribose strand, we're always going to have a phosphate. So phosphate is PO4 at the 5' end hanging off by itself. So there's our 5' carbon, there's a phosphate on it. So again, our 1, 2, 3, 4, and 5' carbons. And the DNA strands are the deoxyriboses are linked together in a 5' to 3' orientation. So we're starting here with this 5' phosphate. And then if we follow this down, you can see that there's another, the next phosphate is linked to the 3' carbon. And so we have phosphate 5' down to 3', phosphate 5' down to 3', phosphate 5' down to 3'. And then at the 3' end of DNA, we typically have a free OH group, a hydroxyl group at the 3' end down here. Okay, so this is the orientation of the DNA backbone. And so right now we're only looking at one strand. If we had another strand here, like we did here, right, you can see here's our 5' end. We're coming down here to the 3' end. But on this strand, we're now looking at the 3' end down to the 5' end. Okay. And so knowing your orientation of your DNA is always an important thing. And again, they're opposite. So like I'm showing you here, here's our deoxyribose is all pointing 5' down to 3'. This little diagram, they actually show a phosphate on the 3', but generally you can think of 3' having an OH group down there. And not, you know, so this side is 5' to 3', this side is 3' to 5'. And that can get a little bit confusing. But generally, when we're talking about DNA sequences, I'm going to be having you play with DNA sequences in a couple of weeks. We typically interpret them as the 5' to 3' direction, because if we had to deal with both sets of base pairs when we worked with DNA strands, it would get really confusing. So essentially, when we work with DNA, we sort of ignore one of the strands, because A will always pair with T, and T will always pair with A, and G will always pair with C, and C will always pair with G. If we know the 5' strand, we automatically know the 3' strand. And so we can basically just work with the 5' strand when we're working with DNA sequences. So we're going to be working with E. coli, and that's sort of represented in this little generic representation of a bacterium here. Genomic versus plasmid DNA. So everything that's alive and some things that are not alive, like viruses, has a genome. And so the genome is the main information that helps determine what an organism is, right? So like I said before, bacteria, the E. coli, have 4.6 million base pairs of DNA that help determine what an E. coli is. And that's its core function. It needs its genome in order to exist as an E. coli. Humans have 3.2 billion base pairs of DNA. That's the core information that helps determine what we are. And I don't mean determine, that makes it sound like genetic determinism, which I don't agree with. But the core information's there. Life has to read the information off of the DNA in order to exist. But all of the core information is in the genome. Bacteria can have little tiny circular pieces of DNA called plasmids inside them that sort of coexist. They exist outside of the genome. And typically bacteria are going to have multiple copies. You know, they can be anywhere from 5 to hundreds of thousands of copies of plasmids just kind of floating around in their cytoplasm. And plasmids are kind of cool. They really only exist natively in prokaryotes. So, bacteria are considered prokaryotes. Prokaryote means it doesn't have a defined nucleus. Things from yeast, plants, humans are eukaryotes. We have defined nuclei and our DNA exists inside the nucleus. Prokaryotes, the genome just kind of floats around in the cytoplasm. It has connections out to the plasma membrane of the organism and potentially connections to the cell wall as well. But plasmids just kind of float around inside the cytoplasm, and they can kind of go around. And oftentimes prokaryotes can sort of exchange plasmids. This is one of the ways that antibiotic resistance can be passed from one bacterium to another. There's a process called conjugation where they sort of exchange plasmids. And also, if a bacterium dies and its plasmids kind of leak out, other bacterium can eat them pick them up from the environment and start utilizing them. And so there's little things like antibiotic resistance that can be picked up through plasmids. This is sort of a representation. Oh, just to sort of also make another point here. The, you know, the genomic DNA, like I've said a couple of times now, that the E. coli's genome is about 4.6 million base pairs. Plasmids are really small. So we're dealing with like 5,000 base pairs. So you can see there's not much genetic information here in a plasmid relative to the entire genome. That, so you can kind of think of it like the um, the genome being the operating system on your phone and a plasmid being an app, right? So you need the operating system in order for your phone to be your phone, but you need some little apps to provide some functionality. And the individual functionality is basically what plasmids do. And so this is a structure of a plasmid that we're going to be using extensively this semester. The plasmid name typically will start with a lowercase p, just to mean that it's a plasmid. And so the plasmid that we're going to be working with mostly is called PEGFPN1. The GFP here is green fluorescent protein. The E stands for enhanced green fluorescent protein. And so it's plasmid enhanced green fluorescent protein. N1 refers to the orientation of the GFP, but you can ignore that for now. This plasmid is 4,700 bases long. So again, it's a relatively small piece of DNA relative to genomes. Some of the core parts of plasmids. So in order to exist out in nature, all plasmids need an origin of replication. So these need to be copied separately from the regular genome of the bacteria. So they need their own place to start replication. And this region of DNA encourages DNA replication machinery to hop on there and copy these plasmids. For our purposes, we have a green fluorescent protein in there. We're going to be sticking something, our gene of interest, in here in this thing called the multiple cloning site. This is something that's been engineered to be in a plasmid. That the GSP has also been in the engineered to be in the plasmid. Essentially, what has happened is over the years, people have found plasmids in nature and they've modified them for different uses. Okay, so this one has green fluorescent protein in it. They also need a promoter. And so promoters, we'll talk about them more extensively later on, but the basic idea is that they drive expression of something. So if you just take a piece of DNA and cram it into a bacterium with an origin of replication, that piece of DNA can kind of exist inside that bacterium and it'll get copied and stuff, but it won't actually do anything. And so promoters allow expression of stuff off of that piece of DNA. And so in its default state, when we take this plasmid, and what we're going to do this week is we're going to extract the plasmid DNA out of bacteria. We can take this plasmid and stick it into human cells, and then this promoter will drive expression of the green fluorescent protein. So if we were to take this plasmid and put it into human cells, it would turn those cells green. This promoter only works in mammalian cells. And so this promoter is not active inside the bacteria. And so, once this plasmid is inside the bacterium, they don't express the green fluorescent protein. Okay, so there's a eukaryotic mammalian specific promoter. Down here, we have this thing called an antibiotic resistance cassette. And this one also has a promoter, even though it's not short, but specifically shown there. And this actually drives expression of this gene in bacteria. Okay, so this thing is actually expressed in bacteria. You can see this thing says CAN-R. That means that it's kanamycin resistant. This one's actually interesting in that it also works in mammalian cells for neomycin resistance, which is G418. So we could use antibiotics, but regardless, let's just focus on the bacteria for now. And so what we do is, if we have bacteria that have this plasmid in there, we add kanamycin to their growth media. And what that does is it prevents non-specific bacteria from growing up in their in the culture with them. Okay, so if we have E. coli and we provide kanamycin in their media, then only bacteria that have this plasmid in them can grow up. If you were to just take bacterial culture media and put some E. coli in there, it's possible that the culture could get contaminated with bacteria that don't have these plasmids in them. So that's one of the ways that we use these. And so you're going to be growing up some bacteria, have some kanamycin in the media, and then extract them. One of the fun things about plasmids is I don't mean to trivialize this, but plasmids have served a lot of functions. Our functions, that we're using them in the lab are sort of basic and mundane, but so like Yersinia pestis, which causes the Black Plague, um, actually causes the plague because of a plasmid that, or actually two plasmids that it had picked up during evolution. And the really wild thing that happens here is that, these bacteria, they're evolved from this thing called Yersinia pseudotuberculosis. That by itself isn't really a harmful bacteria. But once it picked up these plasmids, it now causes a disease. And the reason that it causes the disease, or a plague pandemic kind of situation, is that the bacteria can now produce a biofilm. And when they produce that biofilm, this is a flea. The bacteria produce the biofilm in the flea, and it produces it in the digestive tract. And so this is the normal digestive tract. You see there's nice organization. Everything looks real nice inside this flea digestive tract. But when the bacteria starts producing the biofilm, it blocks the digestive tract. And that causes the flea to get really hungry. It prevents proper digestion. And so that makes the flea want to feed a lot, right? And so the bacteria are in here, they're making the biofilm, they're preventing proper digestion from the flea. So the flea will go bite an organism, it'll get a little bit, but then it kind of regurgitates because its digestive tract is full. And so then it has to pop off and it goes and gets another organism, and it does the same thing. And in that process, it's spreading the disease very rapidly because the flea wants to keep hopping off and going and eating other organisms. And so it goes from organism to organism very quickly. That causes the spread of the bacteria. And it's that behavioral change that really caused the Black Plague. It's not necessarily that this bacterium is more dangerous or less dangerous. It's that the spread happens so quickly, and it's all due to the formation of a biofilm on a plasmid. And so that just gives you an example of some plasmids that exist out in nature. With our thing, we're dealing with antibiotic resistance. That's one of the main functionalities that we use. And again, antibiotic resistance out in the world, nature, and hospitals and whatnot is generally a pretty bad thing. But the antibiotic resistance genes that we work with generally aren't useful for medical reasons, so it's not that big of a deal. The two most common ones that we deal with are ampicillin and kanamycin. Ampicillin inhibits cell wall synthesis for the in bacteria. That structure I showed you, bacteria, they have this thing called a cell wall on the outside of their plasma membranes, and basically ampicillin prevents that from happening. Kanamycin inhibits the ribosome, and the ribosome, or inhibits ribosomes. And ribosomes are the things that perform translation. And if you remember my first slide from pretty much every video so far is transcription and translation, right? Translation is what converts the information from RNA into protein. So if you inhibit translation, you basically inhibit life. And so kanamycin inhibits the ribosome, so that basically kills the bacteria. And it's interesting because this one also kind of works in mammalian cells as well. We just need to change the antibiotic. Um, yeah. So how do we extract the plasmid DNA out of the bacteria? So again, we have our little bacteria drawing here. Here's its genomic DNA. Here's the plasmid DNA. Um, so essentially what happens is we take some detergent, and detergents are things that can basically dissolve cell walls. Okay. Um, and so the detergents sort of break open the bacteria, and all the bacterial parts kind of go everywhere. And so that's sort of represented here. It's kind of a neutral pH. Um, the genomic DNA is typically in a lot of contact with, uh, it's often in contact with the cell wall. It's in contact with a lot of proteins and stuff. And so what we can do is then change the pH. And when you change the pH of proteins, it causes them to denature. And that's what this egg is, right? If you've ever sort of cracked an egg open and you see that it's a liquid, but then you heat it up and it becomes solid, that is denaturing all the proteins in the egg. And essentially we're kind of cooking it by changing the pH here. So we're making it really basic. And that causes all the proteins to denature. The DNA that's associated with those proteins precipitates down as well. So all the genomic DNA that's interacting with these proteins gets pulled down, and we centrifuge all this junk out. And we're left in liquid phase with plasmids floating around because they don't really interact with proteins as much. They're not as dense, and all of this stuff's centrifuged out. And so we take this liquid in the supernatant and we pass it over a silica column. We call it a column. It's a little matrix. Basically, DNA can interact with glass silica in the right amount of salt. Salt causes a little bridge between the DNA backbone and the structure of salt. And so if we have a little bit of salt here, we can get the DNA to stick. We can do some extra washes and stuff. And then we can elute the DNA. And actually, this little diagram shows a lot of salt. We actually use very little salt, and that gets the DNA to come out. And so this process is called a mini prep or a mini preparation. There are different scales. We use about 3 milliliters of bacteria. You can use liters of bacteria. And the name for that changes depending on how big your prep is. And so these 3 milliliter, 6 milliliter preps are called mini preps. And so then once we have our DNA, theoretically, we should have some nice clean plasmid DNA in solution all by itself. We need to quantify our DNA concentration. And so for that, we use something called a nanodrop spectrophotometer. So a spectrophotometer is kind of like the 96 well plate reader, the microplate reader that we used last time. Technically, that one's also a spectrophotometer, but the nanodrop is a specialized spectrophotometer, mostly for determining DNA and RNA concentrations. It can do some other stuff, but DNA and RNA, because of their chemical structure, they absorb light around 260 nanometers. Okay, so that's showing up here. So what the spectrophotometer does is it measures how much light gets absorbed at these wavelengths. So we go from around 220, I think, to around 300 nanometers. And what this tells you is sort of a measure. First, it tells you the concentration of your DNA, but also it can give you an indication as to how clean your DNA is. But this absorbance specifically over 260 nanometers, there's a specific relationship called Beer's law that allows you to determine the specific DNA concentration. This is an absolute concentration assay, whereas the one we did last week is called a relative assay. So if we know certain values, we can determine the concentration of things. Okay, so this is Beer's law. This is how it's written out. A equals weird E, B, C. Okay, so A equals absorbance, right? Absorbance is how much light gets absorbed in a certain assay. Skip down to B here for a second. Path length in centimeters. So how far the light has to pass through the molecule or through the sample. And then C is the concentration of that substance. And whatever substance we're talking about is this thing called the big weird E. It's the molar absorptivity. Okay, so for a given compound, if you know, but this is something that has to be empirically determined for every compound. So basically, if you have a mole of that compound and light passes through a centimeter of that compound, you know how much light gets absorbed by that sample. And so we can rearrange all of this, right? We can get the concentration over by itself, and we can bring the E and the B over here to the A side. And we can say, okay, if we know how much light gets absorbed in a really small path length of DNA specifically, right? This would refer to DNA specifically. This would refer to the path length. This is how much gets absorbed. We know what our concentration is. Okay, so essentially, for DNA in a specialized machine like a nanodrop, essentially our absorbance is directly proportional to concentration. And so when we do our nanodrop, we get a curve like this. Okay, and so it's really this absorbance over 260 nanometers that determines our DNA concentration. And luckily, we don't have to do any math on this one. The machine will automatically do the math for us. This spits it out, the absorbance, as OD. Okay, so OD stands for optical density. It's the same thing as absorbance. Okay, so we're going to get this OD 260 number. The machine's going to spit you out a concentration based off of that OD 260. Something else to pay attention to is the ratio of absorbance 260 nanometers to 280 nanometers. So basically, if you took this absorbance here, our y-axis here is our absorbance. If you took this absorbance right here over 260 nanometers, and you took the absorbance here at 280 nanometers, and if you divide that number by that number, you get a ratio. And that ratio tells you how clean your DNA sample is. Okay, so 280 nanometers, right? We mentioned before that there's proteins that, ultimately we want the DNA to be separated from. Sometimes you get some contamination. And so basically, the higher this number, the more contaminated your DNA sample is with protein. And so we want this ratio to be relatively high. 1.9 is perfect. So essentially, close to 2. You want, if this absorbance came out to 2 and this absorbance came out to 1, that's pretty darn impressive, nice, clean DNA. Keep in mind that this thing will also show you other parts. So some solvents and salts and ethanol can show up in this area, proteins mostly here. What we want to see when we look at these curves is really nice sharp peaks around 260 nanometers. There's always going to be a little bit of stuff coming out over here, and that's fine. If our curve is mostly up here and your DNA is showing up down here, that's not a good thing. Ideally, your curve should look something like this red and this green one. And so what you're going to do is you're going to determine your DNA you're going to use the nanodrop to figure out your concentration and your OD 260 280 ratio. And the machine spits all this out for you. You don't actually have to do any of that math yourself. Okay, so for this week, specifically what you're going to be doing is inoculating a 3 mil culture with JM109. I showed you the plasmid PEGFPN1. We also have the plasmid PM cherry N1. It's essentially the same plasmid, except for this one has a red fluorescent protein instead of a green fluorescent protein. Think about whatever color you'd like to clone your gene as, um, whether you want it to be red or not. This should take you about 5 minutes to do. So, schedule a half hour on the slot on the schedule to come in and do this part. So once you inoculate it, you have to let the bacteria grow overnight. Um, it can be up to 2 days, but do not let it go for 2 days. The next day or the day after that, you can come in and do your mini prep. So I would schedule an hour, maybe an hour and a half to do this, because you'll also have to do your nanodrop DNA concentration assay. Um, and then you'll label your sample and put it in the freezer, and then post your little graph of your DNA, with your DNA concentration and your OD 260 280 ratio to the form on Moodle. All right.